Welcome to Biology 1408, Chapter 4, How Cells Obtain Energy from the Concepts and Connections textbook by OpenStax. This chapter will cover topics related to energy production of the cell. By the end of this chapter, you should be able to describe specific metabolic pathways, the laws of thermodynamics that govern those reactions. We will also dive into how enzymes are responsible for making those reactions occur easier. And once we have a good grasp of those concepts, we will explore in detail the process of cellular respiration and fermentation. Let's begin by looking at two pathways of metabolism. Anabolic metabolism is the building of larger molecules from smaller components. This process does require energy in order for this reaction to proceed forward. The second type of metabolism, called catabolic metabolism, is the breaking down of large molecules into smaller components. This process does not require energy, but rather releases energy after the reaction has proceeded. Since we keep using the term energy, it's important that we understand a few universal laws that govern energy in our universe. The first law of thermodynamics states that the total amount of energy of the universe is constant and conserved. That means we cannot decrease or increase the total amount of energy. Rather, the energy is just transformed into another form stated by the second law of thermodynamics. And in biology, we refer to this form or other form of energy as heat. We can measure mathematically the amount of energy and determine it if it's released or absorbed during a chemical reaction by calculating something called the delta G, or Gibbs free energy of a chemical reaction. If a chemical reaction releases heat energy, it will have a calculated negative delta G. Biologists call these reactions exergonic reactions. If a chemical reaction absorbs heat energy, it will have a calculated positive delta G. Biologists call these reactions endergonic reactions. One of the most important types of proteins in living organisms are called enzymes, because they can affect the total amount of energy required for a reaction to proceed to completion. When the amount of energy required during a chemical reaction is graphed, here in a graph shown here, it is evident that the threshold of energy required to make the transition from reactant to product, shown in the blue line indicated here, following along with my pen. This is the threshold of energy required to make products without an enzyme. However, sometimes the threshold amount is just too high for products to be made, and it can only proceed forward with the help of an enzyme, and it lowers the threshold required to make the product, following along in the dashed red line drawn here. This being the new threshold. Enzymes are said to lower the threshold or activation energy for a chemical reaction. Enzymes function by binding at the active site of an enzyme, which fits together like a lock and key to the substrate. Once the enzyme substrate complex forms here, the substrate is then converted to a product through a chemical reaction facilitated by the enzyme. At this point, the enzyme and product complex will separate when the enzyme releases the products, and the enzyme can then be recycled to perform this function again. This enzyme function can be really written in a 5-step process shown here. I would take some time to pause the video to go through these 5 steps and compare those to the image shown in the previous slide. Now that you've had time to look over the 5 steps of the enzymatic function, let's look at how we can actually regulate that function. And it turns out that the function of enzymes can be exploited by pharmaceutical companies to design drugs which inhibit the function of some enzymes involved in events like, say, pain reception. There are naturally occurring inhibitors. However, scientists can design enzyme inhibitors which essentially block enzyme function. There are 2 main classes of inhibitors, and they are called competitive and non-competitive inhibitors. The competitive inhibitors essentially competes for the active site of an enzyme. How a non-competitive inhibitor works is essentially the molecule or inhibitor binds to an alternative site called an allosteric site, changes the shape of the enzyme, therefore changing the shape of the active site, preventing the substrate from binding to the enzyme. As I said earlier, there are naturally occurring inhibitors. However, not every molecule that binds to the allosteric site is considered an inhibitor. Sometimes other molecules must bind to an allosteric site in order to activate the enzyme so that it can bind to the substrate and produce products. Shown here, this is a type of regulation of enzyme function where we can turn on and off an enzyme through allosteric activation rather than inhibition. Regulation of enzyme function happens in many, if not most, metabolic pathways. The body must have a way to regulate how much of a product is produced. Shown here is an example of a metabolic pathway that includes 3 enzymes functioning to produce a product at the end. Once enough of this product has been made, it can stop the action of the first enzyme by preventing it from binding to the original first substrate. And this is called feedback inhibition. At this point in the chapter, we are going to look at a particular cellular process called cellular respiration. In this process, we create ultimately a molecule that is responsible for putting energy into chemical reactions, and we're going to look at the 3-step process of how that energy molecule is created. In the diagram you see here, the process occurs inside the cell, mostly in the cytoplasm or cytosol, and then transfers into the structure called the mitochondrion, which produces the majority of ATP, the energy molecule in question. So let's dive into more detail about how this process works. So cellular respiration essentially uses a glucose molecule to produce the ATP molecule. And over to the right-hand side, this is an example of the ATP molecule here. It has essentially 3 main components. It has a ribose sugar, a nitrogenous base called adenine, and 3 phosphate groups. Hence the name adenosine triphosphate. Cellular respiration can be broken down in 3 major steps and typically only occurs in animal and plant cells. The 3 main steps are called glycolysis, which is the conversion of glucose to a molecule called pyruvate. Glucose has 6 carbon atoms, and pyruvate has 3 carbon atoms. The second step is called the citric acid cycle, where essentially pyruvate is converted to a molecule called acetyl-CoA, which then is used to pass on electrons to electron carriers called NADH and FADH2. These two electron carriers then move on to the third step, or third major step of cellular respiration, called oxidative phosphorylation. They are ultimately used to drive the function of an enzyme called ATP synthase, which makes the majority of ATP molecules in the cell. The overall chemical reaction of cellular respiration, in a very general sense, is the glucose molecule, C6H12O6, and the oxygen molecule, O2, are used to create carbon dioxide, CO2, and the energy molecule, ATP. Hence the name cellular respiration, where you're breathing in oxygen and breathing out CO2. So let's take a closer look at glycolysis in cellular respiration. Essentially, we're going to start with a 6-carboned sugar called glucose, and we're going to go through a series of enzymatic reactions to ultimately produce pyruvate, and during that process we are going to use ATP molecules but also produce ATP molecules. As a net, we produce 2 ATP during the entire process of going from a 6-carboned sugar to our 3-carboned sugar pyruvate. And pyruvate, both molecules of pyruvate, the one here and the second one here, move on to the next step. Now, the process of glycolysis is actually much more complicated than your textbook describes, and I wanted you to see an overview of the metabolic process, the enzymatic reactions that happen during glycolysis. Keep in mind, this is just so that you can see how complicated the glycolysis process could be, but you do not need to know the names or structures of all of these molecules. Instead, refer to the previous slide. But essentially, the idea is we start with our 6-carbon sugar and end up through several processes, a 3-carbon sugar, but 2 of them. So we're going to have 2 times of those. So let's move on to the second step, called the citric acid cycle. And the citric acid cycle essentially is pulling in pyruvate here and going through a series of chemical reactions that ultimately lead into the citric acid cycle. One main thing is that ATP will be made from that cycle. Carbon dioxide will be made through this entire process, and you will have electron carriers that are created. It's very important that you know that there are essentially 4 NADH molecules, a single FADH2 molecule, and a single ATP molecule made from one single pyruvic acid or pyruvate molecule. These electron carriers then move on into the next step, but just so that you can see how much more complicated the citric acid cycle could get, each one of these metabolic steps in the cycle create a new substance and have an enzyme that actually creates or helps with that next movement of that next step. Again, you do not need to know the names of these structures or enzymes. This is just so that you can see how enzymes are used in that process. So, on the last step, called oxidative phosphorylation, we need to move into an organelle within inside the cell, and this is called the mitochondria. And specifically what we want to do is zoom in on a particular area of the mitochondria, taking note of the outer membrane, the inner membrane, the space in between called the intermembrane space, and the space inside the intermembrane called the matrix. If we zoom in on that area, it looks something like this. When we move to a blown up version of the outer membrane and the inner membrane, you can see that there are proteins that are embedded in the inner membrane that are utilized during oxidative phosphorylation. Keep in mind and note that the citric acid cycle has now does occur in the matrix or inside the inner membrane of the mitochondria. And essentially, the molecules of FADH2 and NADH are utilized along with oxygen to perform one task. And here is the task: to move protons across the membrane. Once those protons are moved from the matrix to the inner membrane space, those protons then need to be pumped back over into the matrix. Those protons move and can only be pumped back over to the matrix side of the inner membrane through a protein called ATP synthase. As protons are pumped across the inner membrane to the matrix, an ATP is created. Ultimately, approximately 90% of all ATP created by a cell are done during this oxidative phosphorylation step. So what should you focus on when it comes to the entire process of oxidative phosphorylation, or the third major step? Number one, that NADH and FADH2 drive protons across the membrane using the electron transport chain, which is the proteins that are embedded into the inner membrane. Oxygen, or O2, is required. Then protons are pumped back across the membrane by ATP synthase into the matrix. And 90% of all ATP are made during chemiosmosis, which is that pumping of the protons across the membrane back into the matrix. What happens, though, when oxygen is not present? ATP is created through several different types of processes we call fermentation. Two of those are called lactic acid fermentation and alcohol fermentation. So lactic acid fermentation essentially bypasses the entire process of the citric acid cycle and oxidative phosphorylation and takes pyruvate, or pyruvic acid, if you will, and goes through an entire chemical process to produce a few ATP and uses an electron carrier in the process. It is not near as much of ATP as oxidative phosphorylation, but some is better than none. However, the byproduct of this entire process is the production of this thing called lactic acid. And lactic acid can build up in molecules or organisms that go through lactic acid fermentation. You, as a human being, can actually go through lactic acid fermentation when you're actually exercising at a great level in which you don't have enough oxygen to sustain the entire cellular respiration process. This would be likened to a situation where you're sprinting and are breathing very hard and do not have enough oxygen to go through cellular respiration. Your muscle cells will then move to lactic acid fermentation to get a little bit more ATP, and in the process produce lactic acid, and this makes your muscle sore after doing that heavy exercise. The second kind of fermentation, alcohol fermentation, also bypasses the process of needing the citric acid cycle or oxidative phosphorylation, and essentially it takes pyruvic acid and creates some aldehyde, acetaldehyde, which then is used to create alcohol, and in the process using an electron carrier and produces a few ATP in that process. The major byproduct here obviously is ethanol, hence the reason we call it alcohol fermentation. This is done by some bacteria in some organisms, single-celled or eukaryotic organisms like yeast. So what could we actually as humans exploit both of these particular types of fermentations for our own benefit in the kitchen. So it turns out lactic acid fermentation we can use to make things like kimchi or sauerkraut or even yogurt. And alcohol fermentation we can use to make our favorite beer or wine and even make some types of bread. Now, this particular glucose metabolic pathway, the entire process of processing glucose into ATP, has connections to other metabolic pathways where we can have things like other carbohydrates, amino acids, even glycerols from fats or lipids come into glycolysis, the production of pyruvic acid from pyruvate, and even into the citric acid cycle. So it is possible that other particular molecules and other metabolic pathways can be used in this glucose metabolism pathway. Now, by this time, you should be able to describe metabolic pathways or reactions, look at thermodynamics with regards to specific types of chemical reactions. You should also be able to describe and identify major steps of cellular respiration and its alternative fermentation. So this brings us to the conclusion of this chapter. And now that we have looked at all of the material in Chapter 4, there are a few things you could do next to help you succeed in learning this material. Take a look at the list shown and do as many as you can. Thank you for listening, and I'll see you in the next chapter.